the organizers for giving me the opportunity to present my recent work here. So today I would like to share you some results of my recent project on protein family-specific relationship between histone modifications and transcription factor binding. So in regulatory genomics, one unsolved question that I also keep asking myself is, how do transcription factors, or TFs, read DNA? And the first thing popped out in my mind is DNA sequence, because most TFs, they bind DNA on a 6 to 20 base pairs of motifs. But some motifs, actually, they only bind to less than 1% of putative binding site in the genome. So what are other factors? And with this snapshot of the 3D genome, actually, it provides us more intuition. And as you can see here, so the gray part is the DNA sequence. It wraps around the light blue histones. And then the colorful molecules here are TFs. And in this example, the TFs bind at chromatin accessible regions. Some TFs bind with their cofactors. Some TFs bind with other TFs and function cooperatively. And actually, during the last few years, our lab and our collaborators worked on DNA sequence, DNA shape, cofactors, and cooperativity, and studied their effect on protein DNA binding specificity, both in vitro and in vivo. And now, in our lab, Jerry is working on a database visualizing the protein DNA interaction, and Zupei is working on electrostatic potential. Sadia is working on CpG methylation and see how they determine the protein DNA binding specificity. And for me, I'm very interested in histone modification and trying to figure out what's the relationship of histone modification patterns and in vivo TF binding, and trying to understand the possible mechanism behind. Previously, ENCODE and Roadmap project have already done extensive work and generate large amounts of data showing there's a close relationship between TF binding and HM patterns. And based on that, there are also quantitative models, either predicting TF binding signals from HM patterns or the other way around. But with that, we are still far away from understanding what's the mechanism and the causality, basically due to two limitations. One, we don't have enough structural information helping us to observe the biophysical changes once the histone marks are introduced to histones. Second, and then for that, we also don't know how those biophysical changes affect TF DNA recognition. And then second, we don't have enough in vitro experiment trying to show us the TF binding affinity changes to the nucleosome containing DNA. But we did observe different modeling performance when we add histone modification patterns to predict TF binding specificity. So we asked, can we answer some question in the middle by taking into account the secondary structure of the TFs? Since TFs from the same protein family, usually they have evolutionarily conserved DNA binding domains. So with these similar DNA binding domains, do they also recognize conserved HM or histone modification patterns around their binding site? So to answer this question, we also took advantage of the ENCODE and Roadmap project resources. After some data quality control, we selected 32, 32, and 15 TFs from these 3 cell lines. And for each cell line, we also downloaded 10 histone modification profiles. And since chromatin accessibility is very important in vivo TF binding, so we also downloaded DNA-seq data and amino-seq data for nucleosome occupancy. And then remember, we want to figure out whether there's a conserved HM signature around TF binding site within the protein family. So we first need to define a set of binding site and non-binding site, right? So for binding site, for each TF, we downloaded the PWM from JSPA database and then scanned the ChIP-seq peaks. And for non-binding site, to make a fair comparison, we concentrated on those chromatin accessible regions and then scanned those regions with the same PWM. And at the end, we make sure the non-binding site has matched GC content and sample size with the binding site. So now we have binding site and non-binding site, and then we're going to look at the histone modification patterns around. So here are the results. We did find certain protein families. They have conserved HM signatures. And I'm going to explain this heat map first. In this heat map, you can see in each row, it has a TF, and each column, it represents a HM, histone modification pattern. And then on the right side of the heat map, you can see the protein family names. And then in the heat map, the colors mean something. Like the white color means there's no significant difference of the histone modification between binding site and non-binding site. And more red means the binding site has larger HM patterns, and more blue means the opposite. So in the rectangle, I highlighted 3 protein families. It's because those protein families, the TFs inside actually has consistent histone modification patterns here. And you can see, for example, for MAD, the heat maps, the color inside actually are lighter compared to ETS and BHLH. So we say this MAD family, it has conserved HM signature, but not significantly different compared to ETS and BHLH. These 2 protein families, we say they have significantly different conserved HM signature. For C2H2, which the zinc finger family, which is also a very big protein family, because their TFs inside has divergent protein structures before those proteins approach DNA. So you can see they also have divergent histone modification patterns here. And then these conserved HM signature for protein families, for these 3 protein families are also consistent in another cell line. So then wondering whether this protein family specific conserved histone modification patterns, it can help quantitative modeling of the TF binding specificity in a protein family specific manner. So here we built a L2 regularized multiple linear regression model trying to distinguish binding site and non-binding site, and we record the performance in AU-ROC. So you can see here in the x-axis, our baseline model takes sequence and shape features, and then in the y-axis, HM augmented models can always outperform. And we found for those TFs from protein families, having the conserved HM signature, they outperform most. And then we want to find out in that HM augmented model, what feature can contribute to, how much each feature can contribute to the modeling that couldn't be explained by other features. So we performed leave one feature out experiment. Each time leaving a feature out of the HM augmented model and recorded the performance decrease. So we recorded the results in this heat map. Again, in the heat map, each row represents a TF, and each column shows a feature that was left out. So this time, for example, this first column, those models, they don't have sequence information, and followed by 4 shape features, minor groove width, propeller twist, row and helix twist, and then followed by 10 HM patterns. So in the heat map, the color, more green means more decrease, and more decrease means more important that feature is, right? So, and based on this performance decrease matrix, we grouped TFs in each cell line into two clusters. For example, for the TFs next to the blue bar, it shows larger performance decrease when we left out the HM patterns. And we say those TFs, they bind DNA in a HM-specific manner. And then for the TFs next to the red bars, since here we can see they have a larger decrease when we left out the sequence and shape features, we say those TFs, they bind DNA in a sequence plus shape specific manner. Then we go back to the protein families having this conserved HM signature at the beginning. For example, for MAD, we say it doesn't have a significantly different conserved HM signature. And for C2H2, it doesn't have a conserved HM signature. So for both protein families, most TFs inside, they bind in sequence plus shape specific manner, and this makes sense. And for BHLH family, since we say it has a significantly different conserved HM signature, we can see most TFs inside, they bind in HM-specific manner. So finally, we still go back to the possible mechanisms, how the HM patterns can affect in vivo TF binding. Then we went back to the literature. Those histone marks, they were reported to change the tertiary structure of the chromatin and also nucleosome stability. So we propose that histone modification patterns, they affect the TF binding specificity for certain TF protein families through their effect on nucleosome positioning. So to test this hypothesis, we investigated the nucleosome occupancy profiles for protein families that both bind in sequence plus shape specific and HM specific manner. So expectedly, we found the nucleosome occupancy around binding site, this red color, and it decreased compared to that nucleosome occupancy around non-binding site. But what surprised us is for those protein families that bind in HM-specific manner, the nucleosome occupancy decrease is significantly more compared to protein family that binds in sequence plus shape specific manner. And this indicates there is a close relationship between the HM patterns and nucleosome positioning, and that might affect certain protein families when they, to recognize their in vivo binding site when they approach DNA. So to summarize, we displayed a protein family-specific relationship between TF binding and HM patterns, and we show the contribution of HM patterns to modeling of TF binding is also protein family dependent. And at last, we propose a possible mechanism showing that the HM patterns affect TF binding through their effect on nucleosome positioning. And with this, I would like to thank my advisor, Remo Rose, for his long-time support and comments and suggestions, and also my lab mates for their help and also comments and suggestions. Thank you for your attention.
Really interesting work. I wonder if you could comment a bit on on the causality. Like, do you know it's that the histone marks are, increasing the affinity for particular transcription factors, or could it be that the transcription factors are recruiting histone-modifying enzymes, or perhaps there are other possibilities?
That's a very good question, and our lab has been thinking about that for a long time. But actually, yeah, both cases happen, and like for protein that we study a lot, MYC. In literature, it was said some certain histone marks are prerequisite for MYC binding, but there are indeed some cases that, like pioneer factors, they bind to the DNA that is in the nucleosome, and later recruit some chromatin modifying enzymes and change the histone marks there. Both cases happen. Yeah, we'll study interestedly. Thank you.
Did you guys consider doing similar enrichment kind of analyses with chromatin state annotations, which would take into account more combinatorial aspects of histone modifications?
Yeah, that's also a very good question. Combinatory effects is also very important. Yeah, we don't have much result for that yet, but I'm very willing to talk about that afterwards. Thank you.